All right, so growing bacteria. So how do you grow bacteria? So one of the things that's different about bacteria versus fruit flies, typically you only want to have one or two fruit flies in order to phenotype, or maybe you're doing a huge genetic experiment and you want to look at thousands of Drosophila flies. So you think about things in the terms of the individual. Bacteria, however, they're so small that we're typically not able to look at single individual bacteria. So, something like this is 5 microns in size. So that's, 1/100th of a millimeter is the size of this bacteria. And this bacteria is actually quite large compared to other bacteria. So one of the things we like to do is we have to grow these bacteria in bulk. And we grow these bacteria in bulk using 2 main types of media. So they can either be grown in liquid. So this is an example of a test tube that contains this liquid broth. And so you would grow the bacteria within this liquid broth. Or you can grow it in a petri dish that has this agar. So this agar is kind of like a gel. So it's derived from seaweed, but it's think of it kind of like hard Jello. So it's kind of has that feeling of Jello, but it's much harder than Jello. And Jello is infused with food dyes and sugar. This agar is actually infused with nutrients. So within, it's not just this hard substrate, but within this hard substrate, this gel type substrate, there's also all sorts of nutrients the bacteria can use. And so in this case, the bacteria can actually grow on top of the agar. So you can see these all this yellow, this thick yellow lines. That is all bacteria, as well as these circular dots. So these circular dots are known as colonies. So whenever we refer to something as a colony, what we're referring to is like a single circular dot. So you can see there's a bunch of these circular dots kind of within there. These lines are not colonies. These lines are still bacteria, but because they don't have that dot-like shape, you typically don't use those. So kind of the goal of growing this on this media is to create colonies, which are these circular dots. So the media contains a food source for the bacteria to grow and kind of all the essential nutrients necessary for life. And now, so we're going to further divide this. So we've already divided the types of media into liquid and plates. And now we're also going to divide the media further into rich media and minimal media. So what is the fundamental difference between rich media and minimal medium? And that is the types of molecules that are within there. And so we're going to start actually with minimal media. So minimal media by its name, it's kind of like the minimum type of chemicals that are required for keeping a bacteria alive. And minimal media will depend on the species that you're talking about. So typically we grow E. coli in the lab. So E. coli is a bacteria that you find in your gut. And it likes 37 degrees Celsius temperature. And there is a minimal media defined for E. coli. And so all that's so this is chemically defined media, which means that we define exactly what goes in there. So there might be water within there. Of course, there's water within there. And then there's a large number of essential salts. So salts contain, ions like magnesium, chloride, sulfate, phosphate, various ions that are kind of essential for life for some region. So all of those ions are supplied in the forms of salts. And then there's also a carbon source. So a carbon source is typically a sugar. So glucose, for example, is an example of a carbon source and an energy source. So the sugar supplies 3 things. It supplies carbon, nitrogen, oxygen. So it supplies 3 of these kind of atoms that are essential to life as organic organisms require carbon, nitrogen, and oxygen. And then it also requires energy. And so in order to build one bacteria into 2 bacteria, it takes a lot of energy. And so this energy comes in the form of sugar. And remarkably, that's all you need in order to grow bacteria. All you need is these salts, glucose, and water. And the sugar source can be glucose, it can be galactose, it can be xylose, it can be sucrose. There's all sorts of different sugars that can be used. So from that, the bacteria is able to actually synthesize all of the molecules that it needs for life. So what are these molecules that it needs for life? So these might be nucleotides. So nucleotides are either DNA or RNA nucleotides that are necessary for creating DNA or necessary for creating RNA. It can form all the amino acids. So there's 20 amino acids. And so all of those amino acids can be synthesized from this sugar. And so those amino acids are then used as the fundamental subunits of proteins. It makes fatty acids, it makes vitamins, it makes pretty much everything that you need in order to create a new bacteria. So that's what you do if you want to make it hard for the bacteria. So that's kind of like the most challenging media, where it has to synthesize all of those different things. And so now this kind of motivates the definition of rich media. And so rich media is media where the bacteria does not have to synthesize these things from glucose. So, typically the way you make rich media is you grind up something that was already alive. So you might grind up yeast or something like that. And so within this ground up yeast is all sorts of essential nutrients. So all those nucleotides can be found, all of those amino acids can be found, all those vitamins can be found. So in these conditions, all the bacteria needs to do is import those into the cell and then use those in order to make new bacteria. So rich media, in general, you also don't know what's in it because you're not synthesizing this from scratch. You're not saying I'm going to add, 1 millimolar of biotin, 1 millimolar of glucose, 1 millimolar of alanine, 1 millimolar of ATP. All you're doing is you're taking some sort of life, grinding it up, and then creating a media out of that. So you don't really know what exactly is in rich media. So this is what we use to grow. So you could have liquid minimal media, you could have liquid rich media, you could have a plate made of minimal media, or you can have a plate made with rich media. So colonies often is what you want to identify. And the reason is that colonies start from a single bacteria. So this colony contains billions of bacteria. Now when you look at it, it's all these bacteria kind of lying next to each other. It looks probably something like this. But originally there was a single bacteria that found its way on this media. That bacteria became 2 bacteria, those 2 divided to 4, those 4 divided by 8, and so on and so forth until there were so many bacteria there that you could actually see them on the plate. So these colonies are genetically identical to each other. So in general, you should just assume that all of the bacteria within a colony are exactly the same as each other. And colonies kind of play the role of the individual. So, you think of the colony as like an individual genotype. So if I look at the different colonies, these different colonies might have a different genotype, but all of the bacteria within the colony share the same genotype. So growing bacteria, so you can go back and forth between liquid and plates. So if you have a suspension of bacteria cells, so you can dilute this liquid culture and then you just put, a small amount of this liquid culture onto the agar plate, and then this is just like a glass rod actually. And so this sterile glass rod you use just to kind of like spread the liquid around so that the entire plate is kind of is soaked in with some of this bacterial suspension. And so you choose a dilution of this bacteria so that there's very few individual bacteria that actually end up on the plate. So there's only 12 or 15 bacteria that actually end up on this plate. And so initially you can't see these things because these bacteria are so small. But again, because these bacteria are undergoing exponential growth, very quickly those single bacteria will become an entire colony of bacteria. So each visual colony that we can see, if we wait 1 to 2 days, each comes from a corresponding single cell that came from the suspension. So serial dilution, so if you wanted to count the number of bacteria in a liquid suspension, so if you had this, you might take 100 microliters and spread it on this plate, 10 microliters and spread it on a plate, and then 1 microliter and spread it on this plate. And so the idea is that, you can count this plate, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there's 12 bacteria per microliter. If you counted this one, you should see approximately 10-fold more. So, I'm not going to count all of these right now. And then if you count this one, you would see approximately 10-fold more. And so this is how you can get you can estimate the number of bacteria that are within a solution. All right, so again, what I said before was, typically when you have a phenotype, you do not, what is a bacterial phenotype? So typically a bacterial phenotype is not something that you can look at and see under a microscope. It's too hard to kind of mount a bacteria on a slide, look at that bacteria under the slide and say, okay, there's some sort of visual feature that I can recognize about this bacteria that's different. Further, once you do that, you kind of lost that bacteria. There's no way to recover that bacteria. If you have a fly, you can look at the fly and then you can grab the fly and use it for further experiments, but you can't really do that. And so the phenotypes that experimenters choose to use are typically whether or not a colony can grow on a nutrient source. So what do I mean by that? So for example, a prototroph, so E. coli is a prototroph. A prototroph can grow on minimal media. So it can synthesize all of the essential organic compounds. A human body cannot, so we can't synthesize all the essential organic compounds. Let's say we isolated a mutant bacteria. So we, one of the things we might have done is mutagenize this bacteria. We've genetically modified it, so some of the genes no longer worked. And so this bacteria is a mutant. And so one way it could be a mutant is that it's no longer able to survive on minimal media. It has lost some sort of enzyme that is necessary for creating one of the essential compounds. And that means that it might not be able to make an amino acid. It might not be able to make alanine. It might not be able to make arginine. And if life is not able to synthesize all of those essential compounds, it's just not able to die. And so you might have a plate that is minimal media. You might have a plate that is rich media. And you have bacteria that we've kind of made a replica plate, so you can imagine like this colony is in the exact same position as this colony. So there's 8, this should be rich media, and this should be minimal media. So there's 8 different individuals that are able to grow if you supply it with everything. But 2 of these individuals are unable to grow on the minimal media. So there's not a colony in the right place right here. And so what that means is that these individuals lack some sort of gene that is essential for life. So how do we determine what is essential for life? So one of these ways we can do that is that we can supplement the media with the essential component. So one example of essential component is arginine. So arginine is a amino acid. And so if we do minimal media plus arginine, then lo and behold, we find one of these bacteria individuals is able to live. And so because that bacteria is able to live, we've identified its phenotype. So we call this phenotype Arg-. So the genotype of this individual is Arg-. And the phenotype that is associated with it is that it's unable to synthesize arginine. It lacks some sort of enzyme that's necessary for creating arginine. And so if we supplement it in the minimal media, we're able to rescue the growth. So this thing is normally unable to live, but if we give it arginine, it's now able to live. This guy is unable to live. We give it arginine, it's still not able to live. So maybe this guy right here is met minus. And so the way we would do that is we would try minimal media plus supplemental methionine. And if it was able to grow, lo and behold, we know that this is a met minus mutation. So all 20 amino acids kind of are examples of that. So it might not be able to synthesize an amino acid. It might not be able to synthesize a certain vitamin. So biotin is an example of an essential vitamin that we need as a cofactor for some reactions. It might be resistant or sensitive to an antibiotic. So this is a common thing that we use. So streptomycin, so strep resistant, strep sensitive. So if the cells are strep resistant, then it doesn't matter if you supplement the minimal media with streptomycin, the cells are able to live. The strep sensitive, if you add streptomycin, the antibiotic streptomycin to the media, then the individuals will die. And then finally, some common ones are sugars. So are they able to use certain sugars as carbon sources? So lac minus and gal minus. So lactose, galactose. So these are different sugars. So your minimal media kind of implicit is that there's some sort of sugar that's within this minimal media. It might be glucose, it might be lactose, it might be galactose. So an example of a gal minus is something that can live on minimal media with glucose, but it cannot live on minimal media galactose, where galactose is the only carbon source. So we're going to have an activity that hopefully helps you become more comfortable with these things. So phenotyping basically involves growing bacteria on different plates. So minimal media contains single sugar source. So this is either galactose, xylose, glucose, sucrose, etc., and salts. So different sugar sources can be used: xylose, glucose, galactose. You can also supplement minimal media with certain things. So an example of that is antibiotics. So you can add, streptomycin, tetracycline, any sort of antibiotic. Different amino acids or vitamins could be added. So you could add methionine, you could add arginine, and you can also add, more than one of these. So you could add methionine and arginine. And then you look to see, does the bacteria live or does the bacteria die? And that is the extent of your phenotyping. And then you can also use rich media. So rich media typically means that it has every single amino acid that you need, every single vitamin that you need. And then you could also supplement that, for example, with antibiotics. And so if you added antibiotics, then strep sensitive or tetanus sensitive or whatever antibiotic you're using, those bacteria colonies would still die. All right, so this is kind of the basics of growing bacteria and phenotyping bacteria. And then you can also do something kind of similar with phages. So phages attack bacteria, and you need a food source for viruses. And so for phages, the food source is actually bacteria that it targets. So E. coli has phages that are able to bind to E. coli cells, inject their typically RNA or DNA into them, and then take over the cell, eventually lysing it and releasing, thousands of new phage particles. So if you want to do this in the lab, that's fine. So you would grow a food source, and then this food source would also have bacteriophage injected in it. So you typically do serial dilutions of the phage so that you find a level of phage where you can create something that kind of looks like a colony as well. So So what if you use a very high amount of phages? So if there are so many phages within the tube, basically all of the bacteria cells die, and then if you plate this on an agar plate, none of the bacteria is able to live. And so all these kind of plaques grow together, all the cells lyse. If you don't add enough virus, then the virus isn't able to take over the culture, and you end up just having a uniform plate of yellowness, and that just means that the entire plate is covered in bacteria. But if you get it right, then there are on the order of 23 viral particles that are within this culture, and all those viral particles are infected. So maybe it's better to say there's 23 infected cells kind of within this culture, this liquid culture. You put it on this plate, and so the bacteria is able to live, except for those 23 individuals that have been infected. So those individuals die, they lyse, and they release particles, and then those particles are able to kind of move a little bit further away and find any bacteria that are next to it. And so this process goes on until the viruses are able to create what's called a plaque. So this is just like a density, a volume of dead cells. So all of these plaques just contain dead lice bacteria. And these viruses we can kind of continue to move out. So each of these viral plaques comes from a single individual viral particle. And so these are kind of the equivalent of colonies. And these colonies can have different phenotypes. These plaques can have different phenotypes based upon how they look. So an example of this is a clear lysis plaque versus a turbid lysis plaque. And so this clear one, it looks like everything is super dark, and then this one, there's still something that looks like a plaque, but it's not quite as dark. And so this is something, for example, that early researchers studied, was the genetics of the phages using this as a phenotype. All right, so that is the end of part 1, and now we will get into genetic exchange between the bacteria.